Good evening, sir.
Good evening, Dr. Pooja. Good evening. Good evening, everyone. So, first of all, on behalf of IAP Allergy and Applied Immunology chapter, I would like to wish every one of you very happy Dashera. And today, on this auspicious day, we are starting a new webinar series. A webinar series which I personally feel that we often understand it very less. It is something which we often deal with. I myself, being an intensivist and allergy specialist, deal with a lot of immune problems using a lot of immune mediators, even in intensive care or in allergy specialty, whether it could be some related to certain interleukins, blocking interleukins, interleukin surge, cytokine storm. But we poorly understand these terms and conditions. We clinicians, we try to, you can say, like, just try to follow an algorithm without probably understanding the things more. So that's why we thought that why not to discuss more about this immunological aspect, and not a very, you can say, like, uh, going very deep into immunology, but whatsoever is very important for the clinician point of view. So it is something like an applied immunology. So in this applied immunology, we will be having 3 webinars, which will be today, tomorrow, and day after, and 2 hours each. And each webinar will be 4 sessions by experts across the globe. So we will be starting today, in which we will be talking about fundamentals. About the fundamentals, obviously, we will be starting with some basics of immunology, and then thereafter we'll be discussing about different antibodies which are available, and these antibodies, what are their fundamental role in the body. We have been always taught that IgE is probably the culprit. It seems that probably IgE is something which is, you can say, like something a bad guy. But is it so bad? So we need to really see that why it is produced in the body and how it is produced in the body, and then thereafter other cytokines and what is the role of those inflammatory markers in some sort of inflammation, whether it is allergic or infective or any other type of inflammation. And then whenever we are ordering a particular test in the laboratory, so which particular patient, which particular child is the right patient, what is the right time, and what is the right test, and how to interpret them. So all these things are really, really very important. We'll be discussing about fundamentals today. I am once again very thankful to all my uh, faculty galaxy, I will say, that we have got, you can say, like, so great faculties with us across the globe. And to moderate them, we will be having Dr. Mangai Sinha. So I would like to invite my colleague, Dr. Mangai. She is a senior pediatric allergy specialist working currently at Navi Mumbai. So, Dr. Mumbai, thank you for Dr. Mangai, thank you for accepting our invite. And please start your session. Thank you.
Thank you, sir. Um, First of all, a very good evening to all of y'all and happy Dashara, everyone. Uh, like Dr. Neeraj said, today we will be having the day one of our Applied Immunology webinar series, which is organized by the IIT Allergy and Applied Immunology chapter. Just like Sir said, um, we know so much about the allergy and the immunological basis, and we always think IgE is the culprit. But in the recent years, there's been multiple research and huge advances which has led to the understanding of the disease right at the molecular and the immunological level. Today, we have experts who will demystify this for us and make it simple. And um, we will see how this knowledge will help us in our day to day practice. So, we have kept it very basic. Today, we'll be discussing the fundamentals of applied immunology, like what is the basics and how does it matter to us in our practice. Whenever we face any patient, what tests are we supposed to order and why are we ordering it? My sir used to always say that when you order a test or when you want to um, investigate a patient, you should always know why you're doing it. So, our experts today are here to demystify the basics of immunology and simplify it for our clinicians. I welcome all our experts today here. Our first talk today will be on basics of immunology simplified for clinicians, innate versus adaptive immunity. And for this, we have Dr. Shambhu Samajdar, an expert well versed with allergy, immunology, pharmacology, with more than 140 articles in indexed medical journals and numerous book chapters. He has received the Governor's recognition for the ACP India Chapter Research and Publication 2024 and Young Scholar Award 2024. He's a consultant physician with JNM Medical College and Hospital, Kolkata. I invite Dr. Samajdar to please start his talk.
Thank you, madam. At the onset, I want to thank respected Neeraj Gupta sir and our moderator, madam. So let's start today's first talk, that is on basics of immunology. And the disclaimer is I am not an immunologist, but from the perspective of clinical pharmacology and basic allergy practice, I shall try to dissect this subject, and we'll try to cover this broad agenda, the difference or what is innate and adaptive immunity, the structures, the anatomy, the physiology, the key cells and effector mechanisms, how this innate and adaptive immune system interplay, and what are the different types of clinical impact and immune dysregulation if it happens, some hyper, hypo, or misfire. We'll try to cover that. I think this is a beautiful picture. We can see the pink one, that is the macrophage, and this they are trying to control that yellow one, and that is the one cancer cell we can see. So this is why this knowledge is important, and we need to know how, for an example, how the vaccine works so effectively, or why does sepsis become fatal so quickly in some person. So for that, actually, we need to know about clinical immunology. So when we talk about immunology, we need to understand how the pathogen can be eliminated, or sometimes it could have some immune escape in the perspective of infection control. Then when we talk about cancer management, then whether certain tumor can have evasion versus immune surveillance, how the vaccine can harness the adaptive immunity, and when the self-tolerance fails, then the autoimmunity, how it is regulation of immunity can trigger certain diseases, the transplantation, and it can lead to certain graft rejection and sometimes immune tolerance. So when there is something called as missing or malfunctioning components, then we will land up with certain immunodeficiency disorders. So these are in fact the core to immunology. And most important is when we talk about clinical success, it often depends on understanding the balance between immune activation and regulation. So if we want to understand the basic of immune system architecture, then the bone marrow, they are the origin of all immune cells. And we all know the thymus, the T cell generally maturate there in the thymus. And when we talk about the secondary immune organs, the lymph nodes, where the antigen presenting hubs, we can say they are the antigen presenting cell hubs. And of course, the spleen is important, especially when we talk about blood-borne antigen filtration. Coming to the important immune cell lineages, if we can divide it in lymphoid lineage and myeloid lineage, generally this is we can say the B cells, the lymphoid lineage, which ultimately tells to produce plasma cells, the T cells, be it the CD4 or CD8 or the natural killer cells. And when we talk about myeloid lineage, the macrophages, the dendritic cells, the neutrophil, eosinophils, etc., there. So generally, if we want to understand the difference between the lymphoid and myeloid, the lymphoid generally they are going to have some adaptive process or surveillance process, and myeloid they are the first line. They are the inflammatory defense for us. And if we talk about the anatomy of immune system, be it organs, some cells, some molecules, so when we talk about different organs, they are coming with tonsils or adenoids, thymus, lymph nodes, spleens, the pear pouch, the appendix, the lymphatic vessels, and of course the bone marrow. When we talk about the cells, then the lymphocytes, the T lymphocytes, we all know the for allergy, the T lymphocytes are the master cell, then the B lymphocytes, plasma cell, natural killer lymphocytes, monocytes and macrophage are important part, and of course different granulocytes like neutrophil, eosinophil, and basophils. And when we talk about molecules, there are antibodies, complement system, cytokines, different interleukins and interferon. So when we talk about immune system, be it innate and adaptive, the basic thing is innate immune system are non-specific. They have cellular components and humoral components too, but adaptive immunity, they are the specific type of immune system, and they have cell-mediated immunity or humoral antibodies. It starts with innate immunity components. They are the first line of defense. The barriers, especially if we talk about the physical barriers, the skin, which is a multi-layered epithelium, they have lots of tight junctions, and of course the mucous membrane, the respiratory, in the respiratory tract, GI tract, urogenital tract. And another important part is the chemical barriers, like for an example, producing acidic pH, producing certain lysozyme, defensins, enzymes in saliva, tears, sweat, and we have our commensal microbiota, the microbial barrier, which also prevent the pathogen colonization. So what, for an example, as a clinician, what is important, if we use unnecessary antibiotic, that will cause disruption in that good microbial frames, and we will have increased risk of infection. And classical example is antibiotic-induced Clostridium difficile. So if we again want to understand what are the cellular components of innate immunity, if we talk about neutrophils, they are having the functions like phagocytosis, neutrophil extracellular traps, which help in early infection control, and the macrophage leading to phagocytosis, helping certain production of cytokine release and APC function. The dendritic cells, the antigen capture, presentation to T cell, natural killer cells which kill the virus infected or sometimes tumor cells, non-MHC mediated, and innate lymphoid cells, the ILC, which will also produce cytokines production and mucosal immunity development. This is an mnemonic we can remember the cellular components of innate immunity. No more danger, kill immediately. So no more danger, kill immediately. Neutrophil, macrophage, dendritic cells, then the kill, the natural killer cells, and finally, the innate lymphoid cells, ILCs. Basic mechanism, the pathogen-associated molecular pattern recognition. For an example, it could be a lipopolysaccharide or double-stranded RNA or dsDNA. So recognition by pattern recognition receptors like TLRs or not-like receptors. And then after that, the phagocytosis, for an example, it would engulf pathogens, fuse with lysosomes, and it will destroy the entire things. And also, if it would lead to hyperactivation, very commonly we had faced this during the COVID-19 crisis, the IL-6. Apart from that, the IL-1, TNF-alpha, which always there during the fever or inflammation time. And during the sepsis, we had not also this cytokine storm. And we need certain anti-cytokine therapies, especially while managing this cytokine storm. So this is an example by which we can understand how the signaling cascade, cytokine release, phagocytosis, and APC activation happen. So the pattern recognition receptors, they detect the pathogen-associated molecular patterns to trigger the innate immune response. In this particular slide, what we can see, one PAM, in this case, that is dsDNA, double-stranded DNA, which produced by the pathogen, is recognized by the PRR, the pathogen recognition receptors. In this case, it is CGAS. And that will activate the sting. And here the sting, STING, is the adapter, forms a complex. And that complex production with TBK1, for example, here, then phosphorylates and activates the specific transcription factor here. It is IRF3. And that IRF3 then translocate to the nucleus to induce specific transcription of interferons. So this is in nutshell how the PRR detect the PAM and trigger the innate immune response. So the take-home message for innate immunity is it is a rapid, non-specific, but it is essential for initial defense, while it can also trigger the adaptive responses. Coming to the adaptive immunity components, the core cell types, the T lymphocytes, the CD4 T helper cells, which recognize antigen on MHC2, which is present in the antigen presenting cell, and orchestrate the immune response via development of different type of cytokines. It could be Th1, Th2, Th17, or Treg cells. Coming to the CD8 plus cytotoxic T cells, they recognize antigen on MHC1. So CD8 means MHC1. And they kill the virus infected or tumor cells directly. Coming to the B lymphocytes, which recognize the native or unprocessed antigens, differentiate into plasma cells and produce different types of antibodies, IgM, IgG, IgA. And in case of allergy, we know there will be classic class switching, and that will produce IgE instead of IgG. So some become also memory B cells. So when we talk about antigen presentation and MHC molecules, this chart is very important to understand, to remember. When we talk about MHC1, that generally present to CD8 T cells, cytotoxic response, and the source of antigen could be endogenous, for an example, certain tumors or certain virus. And MHC2, the CD4 T cells, the helper T cells, generally they will have a source of antigen exogenous. It could be some bacterial or phagocytosed. And for an example, when we talk about APCs, the antigen presenting cells, they are generally the dendritic cells, macrophages, and they require for T cell activation. And also we need some co-stimulatory signals. For an example, we need ligands CD80, CD86. This is an example. And when we talk about memory generation, which is extremely important for lasting immunity, the central in memory, the location is in the lymph nodes, and their function is rapid recall upon re-exposure. And when you talk about effector memory, they're generally located in the peripheral tissues and immediate action at the infection site. So B memory cells, there is persisting long-term and quick antibody production, and generally vaccines, whatever we are getting, they are exploiting this mechanism for lifelong protection. This is an important concept we need to understand. The clonal expansion and memory formation, the first step, the antigen presentation to the naive T or B cells. So the antigen presenting cells, for an example, it could be dendritic cells or macrophages, they will process and present the foreign antigens via MHC1 to CD8 cytotoxic T cells or MHC2 for CD4 helper T cells. Now, the B cells, unlike T cells, can bind the native antigens directly via their surface B cell receptors, and there we need activations through co-stimulatory signals. And generally, why it is clinically important, if there is certain defect in MHC molecule or co-stimulation, that will lead to immunodeficiency or transplant tolerance issues. The next step is the activation or clonal expansion. Upon recognition, the antigen-specific T or B cell become activated and undergo rapid proliferation, which is a process called as clonal expansion. And there could be thousands of identical daughter cells that all recognize the same antigen. This step actually ensure the amplification of the immune response, and we can say that interleukin 2, or in case of allergy milieu, the interleukin 4, 5 is very important. They are actually telling the B cell to have the class switching. So we'll get more IgE instead of IgG. And step 3 is the effector differentiation. So CD8 T cells, they become cytotoxic T lymphocytes, and that kill infected cells immediately. So CD4 T cells, that will become the helper subsets, for an example, Th1, Th2, Th17, or Treg, that actually modulate the response. And when we talk about B cells, they will differentiate into plasma cells, and that will produce large amounts of antibodies. So what is important for us? The effector cells are short-lived but crucial for immediate pathogen clearance. And finally, the memory cell formation. A subset of activated B and T cells, they will become the memory cells instead of effectors. So these long-lived cells will present in secondary lymphoid organs. We had learned that is lymph nodes, it could be spleen, and also in the peripheral tissue, especially when we talked about effector memory T cells. So as we had already discussed, the central memory and the effector memory, B memory cells generally rapidly redifferentiate into plasma cells upon re-exposure. So what are the important clinical points? Vaccination, that their efficacy generally depends on the strong memory cell formation. When you talk about cancer immunotherapy, that also aims to harness the T cell memory against tumor antigens. And we had a lot of autoimmune disease. Autoimmune memory cells may perpetuate chronic inflammation, the rheumatoid arthritis, the multiple sclerosis, and immunosuppression in transplant medicine. That also a huge important subject that aims to suppress the clonal expansion. This is a classic picture from our Goodman-Gilman. What we can see, the antigen presentation and T cell activation, the first stage, and there the APCs generally capture those tumor antigen via the MHC and T cell receptor, the first handshake. And the co-ligand, the CD28B7, for an example here, their interaction provides the co-stimulation for T cell activation. And then the immune checkpoints are also important here to note. Here in this picture, we can see the CTLA4, and this CTLA4 will compete with CD28 for B7 purpose. So that CD28 and B7 interaction will not happen. CTLA4 will play, and that will suppress the activation. Here lies the PD1. Now, this PD1 on T cells, they will bind to those PDL1 or L2 on APCs or tumor cells. That will also dampen the response. Now, the tumor immune evasion happens. Cancer cells upregulate this PDL1 after this interferon gamma signaling, and this inhibits the cytotoxic T cell function in the tumor microenvironment. But what we can have, if we have something which can block those checkpoint inhibitor therapy, for an example, the blockade of CTLA4, that like ipilimumab, which enhance the T cell priming in the lymph nodes, or if we can block that PD1, PDL1 part, like nivolumab, that sustains the T cell function in the tumor. So that will restore the T cell cytotoxicity against cancer cell, promote cytokine release, clonal expansion, and long-term anti-tumor immunity. Coming to certain differences between innate versus adaptive immunity, I think this we had learned from our undergraduate days. The specificity for innate immunity is broad. It recognizes common pathogen patterns, whereas adaptive immunity, it is very much antigen specific, highly precise. The memory for innate immunity it is none except certain trained immunity. But for adaptive immunity, yes, it is there. And long-lasting immunological memory and the vaccine generally play in that part only. The timing, it is immediate type of immunity, the innate immunity within minutes to hours, but adaptive immunity is delayed, like days to weeks. When you talk about cells, primarily innate immunity is the game by neutrophil, macrophages, dendritic cells, and natural killer cells. Whereas the adaptive immunity, there is a B cell, T helper cells, toxic T cells are important. Receptors like pattern recognition receptors are important for innate immunity, whereas in case of adaptive immunity, the B cell receptors, T cell receptors, we can say. The evolution, the innate immunity is ancient, present in all multicellular organisms, and adaptive immunity is mostly vertebrate-specific innovation. So what we can say, when you talk about innate immunity, they are the first responder, critical in early infection and inflammation, whereas adaptive immunity, they are the specialized defense system. They are helping us in case of vaccination and sometimes in problems like transplant rejection or autoimmunity mediated disorders. Together, they form a synergistic system. So innate triggers and adaptive system refines and remembers. So innate immunity acts fast but general, while adaptive immunity is slow but precise and long lasting. So interplay, the bridge is also important. How innate immunity activates the adaptive immunity. The first important part, we need to understand this dendritic cells, which is a professional bridge, we can say. These sentinels of innate immunity, which is the capture, process, and present the antigens and migrate to the lymph nodes to activate the naive T cells. And they will display the processed antigen on the MHC molecules. And there are four important signals which required for T cell activation. The signal one, the antigen recognition. For an example, MHC1, they will talk with CD8 T cells. Then the MHC2 will talk with the CD4 T cells. Now, there is a role of co-stimulation. Already we had talked about the co-stimulators and the co-stimulatory ligands which are present in dendritic cells. For an example, the T cell, there will be CD28, whereas in the dendritic cell, the B7, CD80 or CD86 will be there. And there are certain factors, for an example, which could prevent the T cell energy, so non-responsiveness. And then the signal 3 means the cytokine milieu. After the handshake, there will be some response. The cytokine milieu will get, the dendritic cell will release the different types of cytokines depending on the handshake. And that could be interleukin 4, 5, for an example, in case of Th2 mediated response. And then they will direct the T helper differentiation. So whether we'll get Th1, Th2, Th17, or Treg. And finally, signal 4, if we talk about, they are the metabolic signals. They will, they will, in the terminology of nutrient availability, for an example, having some glucose, amino acid, our gut microbiome, they are the place where it could happen. The gut microbial responses, it could be co-regulated by different pathways like mTOR or immune checkpoints. Why it is important for us clinically? Yes, we talked about checkpoint inhibitors, anti-CTLA4, anti-PD-1, L1. The block inhibitory signals, they will sustain the T cell activity, and there are different type of cancers where these are being used. And of course, this is something which actually have the revolution in the therapeutics of cancer immunotherapy. I think this is a very interesting, important step, because we all practice allergy, and I just want to highlight this picture. And this is a classic example how type 1 hypersensitivity happens and how the antigen presenting cells, they are playing their role. So we all know that when some allergen introduced, they are come inside the tissue due to the defective epithelium barrier, and therefore the allergen will come inside the tissue, and there are antigen presenting cell which will take those allergen here, it could be antigen, and they will enters into the lymph node. And there the T naive cells are waiting, and we can see there will be the first handshake between the antigen presenting cell and the T helper cells. Here the antigen presenting cell, they will give the MHC2, and the T cell have the T cell receptor, and in between the antigen is there. Now also here the co-stimulatory ligands, they are playing their role. For an example, B7 and CD28. Now with this type of interaction, there will be certain cytokines, and those cytokines will ultimately help T cell to differentiate it into Th2 cells. And those Th2 cells will also have an interaction with B cells, and they will also produce cytokines like interleukin 4, 5, and 13. So, and they will tell the B cells to produce or to maturate into one type of plasma cells. And in that plasma cells, instead of having IgG, they will produce IgE. There is a classical class switching that happened. And now that Ig will go out of lymph nodes, and there are lot of mast cells. On those mast cells, the Fc epsilon receptor are sitting, and on those Fc epsilon receptor, the Ig will sit down. Now, when again the allergen will re-enter, there will be a classic clasping, and the mast cell will degranulate, and there will be histamine release, leading to different type of responses. As allergy practitioner, we need to know this thoroughly, that basic mechanism of type 1 hypersensitivity reaction. So when we talk about the innate and adaptive bridge, which is mediated by dendritic cells, which deliver those 4 critical activation signal, we had learned the signal 1 to signal 4. And when we talk about immunotherapies, they will work by removing these inhibitory breaks on this process, unleashing the durable anti-tumor T cell immunity. There are certain clinical case examples, but I think the time is short, but I just go through this. Severe combined immunodeficiency. So there is no functional adaptive immunity. So defective T and B cells, recurrent severe infections early in life, and very low CD4 T cell count. So we need bone marrow transplantation or certain gene therapies. For an example, sepsis, overactivation of the innate immunity via PRRs, example, toll-like receptors, and they will trigger cytokine storm. So we talked about ILC. So CRP will be high, the procalcitonin level will be high. So we need antibiotics, we need certain immunomodulation. And for an example, in certain case, we had used ILC blockade, especially in COVID-19 related cytokine storm. And we talked about cancer and immune evasion, where we understand how the tumors suppress the innate responses, decrease the NK cell activities or antigen presenting cell activity, upregulation of those PDL1 or CTLA. And there are certain markers we had already discussed, and we need therapeutics like checkpoint inhibitors. There are certain key biomarkers we need to also remember. For an example, we had talked a lot about IL-6, the CRP, the marker of systemic inflammation, and of course the CD4 counts, which tells us about the adaptive immunities status. So clinical immunology that spans deficiency, overactivation, and immune evasion, each with distinct biomarkers guiding diagnosis and therapy. So to summarize what we had learned, innate immunity, they are the fast, no memory. When we talk about adaptive immunity, they are targeted, powerful, and they will remember. Their crosstalk is essential for immune control and therapy. And of course, the clinical markers and interventions must target both arms. So today we are celebrating Dussehra, and immunology and Dussehra, there is a shared relation. So it trimmed the good over evil. So in immunology also, we had understand how this mirrors the immune system battles, the innate and adaptive immunity. They are the Rama's army, and we need to prevent the pathogens and tumors. And that is why Dussehra, just like Dussehra, which teaches us about resilience and vigilance, the immune system thrives on balance, coordination, and memory. So thank you. So like the victory of Dussehra, clinical success lies in empowering the immunity to overcome the disease. while keeping balance between attack and regulation. Thank you very much.
Thank you, sir. It was a wonderful talk, and I really like the way you linked today's auspicious event of the Shera, the festival that uh, is about triumphing over the evils, and how you related that with the way the immunity or the immunological system of our body does the same and protects us from all evils and all pathogens outside. It was a wonderful talk, especially talking about the innate and adaptive system in detail, the crosstalk that happens between them, and the entire cascade and all the important cells that are involved in this cascade. Uh, we will take the questions towards the end once we finish all the talks. Thank you so much, sir, for being with us here.
Thank you, madam. Thank you.
We now move on to the next session, which is on understanding antibodies and immunoglobulins. That is, to see the relevance of IgE, IgG, IgA, and IgM. And to explain all of this to us, we have an expert, Dr. Umair Bagir. Welcome, sir. Dr. Umair is a physician scientist, a researcher with excellent knowledge on diseases related to immunity, that is, inborn errors of immunity, with special interest in computational biology and its translational aspect. He has 39 publications and has rendered numerous talks in national and international forums with a long list of awards, including the G.P. Talwar Young Scientist Award by Indian Immunology Society, the ICMR Nurturing Clinical Fellowship for Pursuing PhD, and various awards in quiz and paper presentations in all the conferences that he has attended. It's a pleasure to have you here, sir. Dr. Umair, welcome, sir.
Thank you. Are my slides visible?
Yes, they are.
Okay, thank you. So at the outset, I would like to thank the organizers for giving me this opportunity. My topic for the day is understanding antibodies and immunoglobulins and its relevance in the clinical practice. So I'll be going through as a sir in the previous talk has already shown us the different innate and adaptive components of the immune system. I'll just be briefly touching upon the B cell maturation, the development of the B cells, what is the structure of the antibodies, different function of the antibodies, what are the different immunoglobulin subtypes, different immunoglobulin deficiencies, specific antibody deficiencies, and a note on IgG subclass. So we all know that before birth, the B cell generation happens in the yolk sac and fetal liver and the fetal bone marrow. In the adult life, in the postnatal period, the B cells they mature in the bone marrow. And with the advent of the next generation sequencing, now we know that different genes are responsible at each and every step of the B cell development, rather for that matter, any cell development. These genes primarily can be categorized into three categories. One is the transcriptional factors like the IKZF1 or the IKZF3, and also known as the Icaros or the Alios. This name has come from the faces of the Greek god, the recombinase complex or the RAG1 and RAG2, and the protein tyrosine kinase. Now we know that the transcription factors and the recombinase complex, if there is any mutation in either of the genes responsible for these, it may cause combined immunodeficiencies, whereas the BTK causes a block in the early B cell development, leading to X-linked agammaglobulinemia. The next is a very busy slide. I'm sorry for that, but in this one slide sums up the B cell development from bone marrow to the peripheral blood, and then moving on to the spleen and the lymph nodes. The hematopoietic stems and precursors they go on to develop the pro B cells where the VDJ recombination takes place, and because of the RAG gene, the V gene segment it rearranges and it completes the VDJ-H chain by the early pre-B stage. Between the early pre and the late pre-B stage, a small circular component also comes off. that is the kappa receptor excision circle, which can be used for diagnosing early diagnosis of B cell deficiencies, which is like TREC, that is the T cell excision receptor circle, which are used for screening of severe combined immunodeficiencies. The early pre-B cell, these cells they express the CD10 and the CD19, and they halt. These are the cells in which the RAG expression is halted to induce the proliferation, and the light chain rearrangement takes place. After that, an immature B cell is formed where the light chains they replace the surrogate chains, forming a surface IgM, that is the B cell receptor, which egress from bone marrow and they move on to the circulation. In the circulation, the immature B cells they enter the spleen as transitional B cells, and these cells they receive signals from BAF receptor, and they can mature into two types. They can either become the marginal zone like B cells, or they can become the follicular B cells. The marginal zone B cells they develop into the short-lived plasma cells, and the follicular B cells which form a T cell dependent, which respond to T cell dependent stimuli by antigenic activation. They enter the germinal center, and the activation it triggers the uh, class switch recombination machinery. The memory B cell, they change the BCR affinity and the isotype and eventually form the IgG, IgA, IgM, or the IgE, depending on the chain. We all know that the human B cell immunodeficiencies, it can be divided into the early B cell development phase, in the genes that are involved in the class switch recombination, and the genes that lead to terminal differentiation of B cells. The initial genes, these you can see the genes, the SAMD9L, the SAMD9, the FANCI, the DKC1, these are the genes that are responsible for marrow failure syndrome along with the B cell deficiencies. From pro-B to pre-B, the genes that are involved, these lead to absent B cells, and they will have a gamma globulinemia with no B cells, and they are a close differential for bruttosin tyrosine kinase deficiency, which causes X-linked A gamma globulinemia. So if a patient has X-linked A gamma, we are suspecting to have X-linked A gamma globulinemia with a normal BTK expression by flow without any underlying genetic mutation identified on an exome sequencing and other functional assays. We will have to look closely for these genes that are causes of autosomal recessive or autosomal dominant A gamma globulinemia. The mature B cells undergoes class switch recombination, and it requires help from the T cell as has been previously described. And these genes are responsible for a phenotype like an hyper IgM syndrome, where we have a deficit in the IgG production, but the IgM production can be normal. The CD40, the 40 ligand, AICDA, and these are the genes responsible for that. And rest of the genes where we can in the peripheral blood appreciate a normal count of B cells, but has a marked deficiency of IgG as per the age. These patients may be suffering from a common disease called as common variable immunodeficiency disorder. The reason why it is called as common is because cumulatively it is the commonest symptomatic inborn error of immunity. The asymptomatic one being the selective IgA deficiency. Coming on to the structure of antibodies, as we all know, it has the variable region, the constant region, and the hinge region. The variable region, it consists of the amino terminus for both the light chain and the heavy chains, and it consists of around 100 to 110 amino acids that determine the antibody's unique antigen binding specificity. The constant region, it defines the light chain subtypes, and five heavy chain classes are there. The gamma, the alpha, the gamma, alpha, mu, delta, and epsilon. And these are the ones that are responsible for the G, A, M, D, and E immunoglobulins. They mediate the antibody's effector function, that is the binding complement, cell receptors, etc. And this hinge region, it is of two types. In the IgG, IgA, and the IgE region, it is proline rich and it provides flexibility, whereas in the IgM and the IgE region, it lacks hinge region and instead it contains an additional constant domain in the middle of the molecule. So this is a gross structure of the antibodies that are of different types. We all know this classical image from Robin's textbook of pathology. We all know how the B cells proliferate, they differentiate into either affinity maturing, which will later on form the memory B cell. They will undergo class switching and they may produce antibody secreting plasma cells. So these antibodies, they are known to neutralize microbes and different toxins. They can help in opsonization and phagocytosis. They can cause antibody dependent cytotoxicity, and with complement activation, it can cause lysis of different microbes, phagocytosis, or inflammation. The immunoglobulin G is the commonest immunoglobulin that is present. It provides majority of the antibodies, and these antibodies help in fighting infection against different pathogens. These are the only antibodies that are able to cross placenta, and it has an important clinical implication. The IgM is the immunoglobulin that is produced in an acute stage. It eliminates pathogens in the early humoral response. The IgA is the secretory antibody. It is found in mucosal areas, gut, respiratory tract, and it is a challenge when the patients with common variable immunodeficiency have very low or absent IgA because it cannot be replaced. The IgD, it is the antigen receptor not on B cells that have not been exposed to antigen, and it activates basophils and mast cells. The IgE, and it does not need any introduction for the robust gathering of allergists, that it binds to allergens and it triggers histamine release, which also has an anti-parasitic function. So the five immunoglobulins have different composition depending on the heavy chain and the light chain. Depending on the underlying subclass, the IgG has four different type of gamma chains, the 1, 2, 3, and 4, and these eventually lead to the four subtypes of IgA, IgG. This is the diagram from Fleischer's immunology textbook, and it shows age-related changes in the serum concentration of immunoglobulin. It is very important that we know that the maternal immunoglobulin IgG, it crosses the placenta and it provides immunity to the child for up to 6 months of age. And it is at that time where the child's immunoglobulin will take up the function of protecting the individual. And this is the reason why most of the patients with antibody deficiencies, where only the B cell functional effect is there, like the X-linked A gamma globulinemia, these patients present after 6 months of age because maternal IgG has been protecting them. So, also we need to keep into our mind that as the maternal immunoglobulins fall, sometimes the child's immunoglobulin may not rise up to 1 or 2 years of age, and it may be a condition called as transient hypogammaglobulinemia of infancy. So we need to repeat the child's immunoglobulin levels so that we are able to exclude this disorder, and it may and these patients may present with bacterial infection in the early infancy, in the late infancy. Coming on to two case scenarios. Suppose we have a 10-month-old boy on one end and a 10-year-old boy on other end. Both the children have had recurrent pneumonias. The 10-month-old boy has failure to thrive and the 10-year-old boy has poor weight gain. When we think about differential diagnosis for these two cases, we need to consider the age at onset. If it is a 10-month-old boy, the chances that this child may have antibody deficiency due to either combined immunodeficiencies, or X-linked A-gamma globulinemia. But a child who's a 10-year-old boy and who was essentially asymptomatic up to 2 to 4 years of age, here the differential diagnosis we may consider common variable immunodeficiency disorder, specific antibody deficiencies, or IgG subclass deficiencies. The differential changes depending on the age of onset, and as we'll see in the upcoming talks, what are the different tests that can be ordered to diagnose the same. This very nice review by Rebecca Marsh et al., where they have given the pattern of immunoglobulin deficiencies. If the patient has only IgA deficiency, then it might be a case of either IgA deficiency or it may also be associated with some IgG subclass deficiency. If there is a pan-hypogammaglobulinemia where the G, A, and M are absent, it may be A gamma, it may be X-linked, autosomal recessive, autosomal dominant, or also associated with combined immunodeficiencies because this A gamma is there because there is no help from the T cells. If the IgM levels are normal or elevated and A and G are low, then we may think about hyper IgM syndrome where we have both autosomal recessive and X-linked conditions. If the A and M are normal and only IgG is low, then it can be an evolving humoral deficiency, it can be a transient hypergammaglobulinemia, or even may be seen in some cases of combined immunodeficiencies. If all the immunoglobulins are normal and these children they were strongly suspected to have an underlying immunodeficiency, then we need to roll out specific antibody deficiency or other humoral and combined immunodeficiencies where these immunoglobulins that are produced may be functionally abnormal, but the levels compared to the age-wise reference range are normal. With the advent of repertoire sequencing, there are multiple studies that have been done over the years where people have evaluated the V gene, the D gene, the J gene, the CDR length, and the repertoire diversity with the somatic hypermutation. This was done on both the DNA-based sequencing as well as RNA-based sequencing, and you can appreciate that most of these antibody deficiencies as well as combined immunodeficiencies, they have decreased somatic hypermutation. Their repertoire diversity is decreased, especially in CVID, and they have longer CDR3 length. And these subtle defects can be seen in a broad category of antibody deficiency. Coming on to the specific antibody deficiencies, this is a subtype of inborn errors of immunity that comes under the predominantly antibody deficiencies, where most of these children can be missed because they have comparatively milder phenotype. We all know that the specific antibody response, it can be assessed by evaluating the protein vaccine response or the polysaccharide vaccine response. The protein vaccine response, they play a role in identifying T cell dependent antibody production, whereas the polysaccharide response, the polysaccharide vaccines, they help in assessing the T cell independent pathway, and these are the ones that are helpful in diagnosing specific antibody deficiency. In India, we all have easy access to evaluating the anti-tetanus levels, but there is a challenge when it comes on to as evaluating polysaccharide vaccine response. The WHO protocol for evaluating anti-pneumococcal response is an elaborate protocol. It's a very lengthy, robust, systematic protocol where we need to evaluate at least 13 or 23 different serotypes, and then the baseline, the change in the baseline versus the follow-up after 1 month after vaccination, the follow-up response for the specific IgG against the particular serotype is at least twice, and these are available only on research basis in our country. So we all are also aware of isohemagglutinin. These are the natural anti-A and the anti-V antibodies that are present in our blood, with the exception of patients who with the exception of patients who have AB blood group. This is a review where the patients with specific antibody deficiencies were evaluated, and you can appreciate that the infection only phenotype is present only in minority of these cases, and majority of the patients also have non-infectious complications like autoimmunity, ITP, EVANS, rheumatoid arthritis, alopecia. So if the patient has infective phenotype or chronic lung disease, where we are also suspecting an underlying antibody defect and we have ruled the common antibody deficiencies, we need to evaluate these children for specific antibody deficiencies. Coming on to the IgG subclass. So these subcategory, these are the patients where the IgG level can be normal. As you all can appreciate, IgG1 is the commonest subclass for IgG, and it is essentially covering most of the total percentage of IgG. So what experts say that if a patient has low normal IgG, it is very well likely that the patient may have normal IgG1. Because these are comparatively expensive investigations, we sometimes may have to prioritize what test can or what test should be done. The IgG2 subclass, it is associated with specific antibody deficiency, and it is reported to be lower even in normal. You can appreciate that it is effective against the polysaccharides, but it is not a alternative for specific antibody deficiency, and the anti-typhi or anti-pneumococcal vaccine response needs to be done. IgG3 is a small percentage of IgG, and it can cause IgG subclass deficiency. IgG4 can also be related to the IgG4 related disease, and it can also help in diagnosing those disorders. So, with this short presentation, we have seen the different subtypes, different types of antibodies, the B cell development, how the different genes at different stages are responsible for the B cell development, and how a mutation in either of these genes can lead to different inborn errors of immunity. This is our team at ICMR NIH, where we get referrals from across India for diagnosing different inborn errors of immunity. Thank you.
Uh, Excellent talk, Dr. Umair. Uh, it was really a brush up of our knowledge of the structure, function of the immunoglobulins, the antibody response. I really like the way you started with the basics of structure, and then even you went to the details and even went up to the how the antibody response is given, and the IgG subclass also. So it was like a talk which started simple and then it went to like it just went on increasing. But it was a wonderful talk, and I think all of us will really agree to that. So thank you so much for this talk. Uh, Thank you, sir. We now move on to our next talk, which is on the role of cytokines and inflammatory pathways in allergy and infections. For this talk, we have an expert, Dr. Pooja Srivastava. She is an immunologist and rheumatologist. She's done her MD from PGI Chandigarh and DM in clinical immunology and rheumatology from SGPGI Lucknow. She has numerous publications and is currently attached to Star Clinics Division of Rheumatology, Allergy and Immunology in Ahmedabad, Gujarat. So who better to take us through this journey of cytokines and inflammatory pathways? Thank you, ma'am. Thank you for joining us.
Thank you, ma'am. This has, like, last two talks were really good. Got to learn and refresh a lot. So I'll be covering cytokines. This is something which comes everywhere. We don't know what started, where the cytokine started or the cell started. So there is always this chicken and egg story. But to begin with, what are cytokines? They are hormones of the immune system. They are polypeptide proteins which are secreted by immune cells and non-immune cells, and they help in all the effector and communication system in the immune system. So this is the way how cells communicate. Now, cytokines can have pleiotropic effects, like they may have more than one receptor. The same cytokine can bind to different receptors. They can be redundancy in their functions also, like one cytokine can bind to different receptors. So same cytokine can bind to different receptors. One cytokine, different cytokines can bind to same receptor, and some few of them could have very specific and unique functions. So all kind of combinations are possible. The classification is mostly based on the target receptors. It also depends primarily on the extracellular structures of the cytokine receptors. And predominant, there are 5 types of cytokine receptors. And these receptors are important because when we actually plan to block these cytokines for treatment of diseases, we need to understand the structures and both the extracellular domains and intracellular domains, the pleiotropy and redundancy, and then decide which cytokine to block, how to block, what will be the good and what will be the bad effects of those. And like this cytokine, now if as we in the first talk we saw that dendritic cells innate immune system, it leads to activation of adaptive immune system. And the interaction between dendritic cell or macrophages with the naive T cell uh, leads to development of all different types of immunity: type 1 immunity, type 2, type 3, type 4. So development of Th1, Th2, Th9, Th17, Th22, and T regulatory cells. But what decides that when a naive T cell comes in interaction with the antigen, in what way it will develop? So that is decided by the different transcription factors and the cytokine milieu which is present there. So like we see, if there is IL-12 and STAT4, it will go on to develop into Th1 type 1 immunity. If it is IL-4, STAT6, and GATA2 as a transcription factor, then it will go to type 2 immunity. So it is there. So cytokines basically help in programming the whole of how the effector immune response happens. So there is a cytokine cellular organ network in the body, and functionally, the cytokines can be classified as either pro-inflammatory or anti-inflammatory. Pro-inflammatory ones are predominantly interferon gamma, perforin, granzyme, TNF alpha, TNF beta, and anti-inflammatory ones are classically IL-10, TGF beta, which helps in development of T regulatory cells. They can help in direct cell migration and adhesion so that the cells can migrate to the site of infection. They can help as hematopoietic colony stimulating factors in development of hematopoietic cells. They can help in development and wound healing by migration of stromal cells at the sites of injury, and they can have regulatory roles like maintaining immune system. So it ultimately helps in all ways. And by the means of their function, they can be called with very different names, like chemokines. The chemokines are those cytokines which have function to attract cells to the site of infection via chemical signals. Interferons, these are a subset of cytokines that prevent viruses from multiplying and also helps in disrupting virus transmission. Additionally, they stimulate NKT cells, which also help in antiviral immunity. Interleukins are the proteins that manage immune and inflammatory reactions, most of the pro-inflammatory ones. Lymphokines, these are cytokines which are secreted by the lymphocytes to communicate with either other lymphocytes or macrophages or other cell types. They instruct these cells to move in and assist in the infected region once the lymphocytes have identified that there is an internal or external threat. And TNF, tumor necrosis factor, this is a cytokine which is there to kill. It eliminates cell. Upon its release, TNF can attach to cancer cells, infected cells, and cause their destruction. So overall, this picture gives us a broad view of cytokines. Once the innate immune cells, either dendritic cell or macrophages, they come and interact with naive T cell with the primary MHC TCR interaction as well as CD80-86 interaction, so co-stimulation. And then depending on the cytokine milieu, it will happen that whether if there is IL-12, TGF-beta, the IL-2 is the T cell growth factor and TGF-beta is the anti-inflammatory cytokine. It leads to development of T regulatory cells, which can help in suppression of tumor immunity. It can promote immune tolerance and maintains lymphocyte homeostasis. If there is predominant cytokine is IL-12, then it leads to development of type 1 immunity, Th1 type of cells, which promotes tumor immunity. It is helpful in protecting from intracellular pathogens like tuberculosis, and it can drive autoimmunity. There are a lot of diseases mediated by Th1 immunity. If the predominant cytokine is IL-4, it leads to development of type 2 immune response, development of Th2 cells, which in turn help B cells and lead to production of IL-5, IL-13. And the main function of Th2 immunity is to protect against extracellular pathogens. When it is dysregulated, it leads to allergy, asthma, food allergy, allergic rhinitis, CRS, those kind of symptoms. And once in the development, if there is predominant cytokine is TGF-beta, IL-6 and IL-21, it leads to development of Th17 pathway of immunity. Their role in tumor immunity is controversial, but their main function is to control extracellular bacteria, and they have a major role in control of fungal immunity also. They are also involved in a lot of autoimmune diseases. So overall, what decides here is the cytokine milieu, what is going to happen. Now, this cytokine milieu also decides on the maturation of macrophages, whether if it is classical activation of, it's a naive macrophage, if it is like classically activated in an inflammatory milieu with inflammatory cytokines like TNF alpha and interferon gamma, it leads to development of M1 macrophages, which leads to the production of pro-inflammatory cytokines like IL-1, TNF alpha, IL-6 and interferon gamma, which can lead to wound cleaning, phagocytosis, tissue damage, and leads to an inflammatory kind of milieu. Alternatively, if this naive macrophage is activated by by IL-4 and IL-13, then it leads to development of M2 macrophages, which has anti-inflammatory role. They will lead to increased production of TGF-beta and IL-10 and VEGF, which leads to tissue remodeling and immuno regulation, and thus development of tolerance and development of anti-inflammation. So here also cytokine milieu is important. So today's our talk is on type 2 allergy. So we will be focusing more on how the the cytokines of type 2 immunity like IL-4, IL-5, IL-13, IL-9, how they mediate diseases and how blocking them helps us to treat these diseases. So how does the initiation of type 2 immune response is generally there is some barrier dysfunction because of either allergens, helminths, hygiene products which we use like sanitizers, lot of soap, pollution in the air, nanoparticles, microplastics in our food or processed food, or so these barrier dysfunction leads to activation of alarmins. Alarmins are IL-25, IL-33, TSLP, which are secreted by epithelial cells once there is disruption of the junction between epithelial cells. And these activate in the mucosal tissue, they activate type 2 innate lymphoid cells. And in skin, they activate the dendritic cell. These ILC2 or dendritic cells then stimulate naive T cells and lead to development of activation of T follicular helper cells, which help activate B cell to produce more of IgE, and these will go on to act on the effector cells. So these are the main cytokines and alarmins in type 2 immunity. So this type 2 immunity, is it always bad? No, it is not always bad. It has good effects, like it activates anti-helminth immunity, it augments barrier defense, regulates tissue repair and regeneration, it neutralizes toxins, it suppresses type 1 autoimmune diseases. So once type 2 is more, it will suppress type 1 diseases, and it maintains metabolic homeostasis. But once it is dysregulated, it leads to, it can promote a lot of allergic diseases. It reduces protective type 1 immunity. It mobilizes immunosuppressive macrophages in tumors. It antagonizes the anti-tumor cytotoxic effects which are there. So tumors can sometimes use type 2 immunity to propagate, and it can induce fibrosis. So what decides whether type 2 immunity will lead to protective tolerogenic effects or it will lead to pro-inflammatory signals is the baseline cytokine milieu. So once there is low allergen dose, a lot of alarmin release, lot of inflammasome activation, viral infections, there is altered gut microbiota, it can lead to develop type 2 inflammation. It develops into pro-inflammatory signals. And once it is, there is very high allergen dose, there is normal microbiota, there are a lot of neurotransmitters, vitamin D is sufficient, the metabolites in the intestine are good, the gut bacteria is good, then it leads to development of tolerance and homeostasis. Sorry for the background noise. There is a procession for the Shera going on just outside. So if we see that just tells us about the difference between the developed world and the developing world. In developed world, where there is no or very less microbial stimuli, there are a lot of allergens, food allergens, sting allergens, house dust mite or indoor allergens, and pollens. They lead to development of pro-inflammatory type 2 responses, which leads to allergic diseases. Even the hygiene and the vaccinations, the alum in vaccine, which is used to preserve it, can also lead to development of pro-inflammatory signal. And in the developing world, where there is lot of very high doses of microbial stimuli, lot of helminthic parasites, lot of viral infection, lot of bacterial exposure, it leads to a development of protective anti-inflammatory type 2 responses. So overall, nearly half of the world's population harbors helminth infection or they suffer from allergic disorders. This is half an hour. So overview, what actually initiates the type 2 immunity problems here is the epithelial barrier. Allergens, chemicals in our routine daily use in cosmetics and soaps and washing detergents, sanitizers, and nanoparticles and microplastics, they lead to epithelial damage. Sometimes a lot of skin infections or scratching, or those can also lead to microbial dysbiosis. And less exposure to plants leads to decreased biodiversity and opportunistic skin infections. All these lead to damage in the epithelium and release of alarmins. Alarmins, again, it goes by the same pathway, activate Th2 immune response and cause inflammatory response. The protective mechanism, how the type 2 immunity actually protects, is the same cytokines, IL-13, IL-5. Once the immune, if we see this, if our epithelial barrier is intact, the same cytokines, IL-13, IL-5, they lead to a lot of secretion of mucus, and they help in expulsion of they induce like lot of mucus production in the intestinal mucosa, and they induce muscle contractions to flush off the parasites out of the gut. So weep and sweep mechanism. They also lead to increased eosinophil recruitment at the site of infection. These cells can help kill and expel the parasites. And it also helps in tissue repair. It activates the macrophages and other factors to help repair the damage caused by parasite. However, once this epithelial barrier is damaged, these helminth infections and the particles can enter, and it stimulates the activation of type 2 immunity and allergens. So in our routine day-to-day activity, the toothpaste we use, the soaps and detergents we use, the sanitizers we use, uh, even the alcohol hand rub which we use, they all can damage our epithelial barriers. Many creams and lotions which are harmful to our microbiome, SLS and hygiene products reduces the barrier function and promotes pathogenic germs and allergens. So if we are using it, we have to use it in moderation or minimum only when it's needed. Further, the regular use of PPIs, it leads to gastric acid inhibition and impairs digestion, and again promotes intestinal microbiome development. So antibiotics also disrupt the microbiome, and antipsychotics often take antibiotics. Antipsychotics often people who are with psychiatric diseases who are taking lot of antipsychotics for years together, they also have antibiotic-like effects. So all these can be the initiating factor for type 2 immunity. What we can do to avoid this is increase fiber-rich diet, increase omega-3 fatty acid supplementations, increase exposure to diverse plants and animals can help increase our or improve our microbiome and protect us from these mechanisms. So how these alarmins, we discussed that breakage of epithelial barrier can lead to either because of inherited filaggrin deficiency or because of scratch from pollen, house dust mite, or animal hairs, or secondary infections like scabies, helminth virus, bacteria. They can all lead to break in epithelial barrier and release of alarmins, which will go on to activate ILC2 and in the mucosal cells, and production of IL-5, IL-13, and activation of eosinophils, mast cell, goblet cells, and lead to development of either atopic dermatitis, asthma, CRS, food allergies that we'll see. So overall, type 2 Th2 cells and in type 2 innate lymphoid cells are the main heroes who run the type 2 immunity, and our cytokines, the predominant ones, IL-4, 5, 9, 13, are the main workers who help them to achieve their goals. And above them is the T regulatory cells, we can say the Supreme Court, which if needed, can sometimes hold them. So how this thing happen? This ILCs or Th2 cells, they because of release of lot of pro-inflammatory cytokines, they can further cause epithelial damage. By IL-5, they stimulate eosinophils to release histamine and the mediators from eosinophil, which can cause damage and inflammation. IL-9, they secrete IL-9, which activates mast cell, and IL-13, which increases the mucus production by goblet cells and leads to smooth muscle hyperplasia. And the Th2 milieu also leads to polarization of macrophages to anti-inflammatory type. It depends on exact milieu, whether it will go on to M1 or M2, and that will decide the further cytokine milieu. So once we come to different diseases, the same thing in asthma, what we discussed, the break in epithelium, epithelial injury, release of alarmins, induction of type 2 inflammation. And here what is happening, IL-4 and IL-13 is causing modulation of smooth muscles in the airways. IL-5 is activating the eosinophils to release histamine and other mediators. IL-4 and 13 is causing goblet cell hyperplasia, smooth muscle hyperplasia. So it can, over long time, it leads to increased mucus production, goblet cell metaplasia, hyperplasia, subepithelial fibrosis, which can lead to irreversible airway thinning, epithelial shedding, basement membrane breakdown, increased production of nitric oxide, and smooth muscle proliferation. And these are the exact cytokines which we are blocking in uncontrolled asthmas, the stage 5 of GINA. And it really helps us to control the disease. In same thing in atopic dermatitis, either genetic deficiency in filaggrin or epithelial colonization by Staph aureus, or there is break in epithelial junction, release of alarmins, induction of ILCs or Th2 cells, production of same cytokine, which leads to polarization of macrophages to M2, more Th2 kind of cytokines, release of histamine causing more itch, urticaria, and activation of mast cells, which leads to release of inflammatory mediators, activation of B cells, which leads to more and more IgE production. So the vicious cycle goes on. Simultaneously, it's the IL-31 stimulates the sensory nerve, which leads to increased catching, and that leads the vicious cycle goes on. So therapeutic targets in atopic dermatitis, again, the same cytokines, IL-4 receptor blockade, dupilumab, IL-4, IL-13, that is IL-4 receptor alpha blockade, helps. This Th2 pathway, Th17 pathway, it leads to a lot of acanthosis, skin thinning. So Th17 pathway is also associated with psoriasis. So a lot of like proliferation and epithelial thickening and dryness is mediated by that. So blocking that also helps. And the intracellular pathway of these cytokines are all mediated by JAK-STAT pathway. So lot of JAK inhibitors like abrocitinib, baricitinib, upadacitinib is also shows promising results in atopic dermatitis. Same thing in food allergy. Till there is not much dysbiosis, some bit of infection, some bit of allergens enter, and we are able to, till there is a balance between Th2 and T regulatory cells, things go on. So when the allergens enter only through the intestine, it leads to stimulation of T regulatory cells, and we can manage with the food. However, once there is break in the skin mucosa, or there are sensitized cells even in the intestinal mucosa, which can lead to early type 1 hypersensitivity reactions, can lead to can actually tilt the balance towards development of type 2 immunity. So this dysbiosis can actually tilt from tolerogenic oral response to inflammatory cutaneous response. So application of biologics in allergic disorders we have seen, and there is a detailed discussion on all these cytokines which can be blocked. These three are already available: IL-5 blockade, IL-4 receptor alpha blockade, IgE blockade are readily available. TSLP, the alarmins, TSLP and IL-33 blockade, these are not yet available in India, but are available, has been approved. IL-13 blockade is still in pipeline. IL-9 blockade still in pipeline. This IL-31 blockade is especially so that Th2 cytokines don't activate the sensory nerve and to break the itch cycle. So overall, the same kind of allergens, once they are given in very high doses, the cytokine milieu changes. So once allergen entry is at a very natural high dose or at immunotherapy routes very high dose, what we see is there are anti-inflammatory cytokines. They take over and it leads to development of tolerance, anti-inflammatory effects. So overall take-home message will be the cytokines, exactly if they are the ones who are playing the cards, or it's the effector cell, or it is actually the entire milieu, and how, like if it is gradually presented, it is given at a go, that actually decides the kind of effector response. So we can equate it to cooking. Like same kind of food, if it is cooked by baking, barbecuing, grilling, shallow frying, deep frying, the taste changes. Similarly, the cytokine milieu and how the antigens or the simulatory things are presented at a very high dose, at a very slow dose, as an intermittent, and the milieu around actually decides the type of effector responses. So the take-home message will be the initiating factors of type 2 immunity can vary with specific tissue microenvironment, that is the cytokine milieu. Epithelial cell are of importance, and the break in barrier is really what is the trigger in most cases. As the initiation of type 2 immune response progresses, a variety of innate cells, including macrophages and granulocytes, and then Th2 cells, the lymphoid innate, and then the adaptive, they take over and they shape the development of type 2 immunity. Thank you.
Thank you. Thank you so much, Dr. Pooja. I think before I go on to the talk, I must say I really loved your slides. They were very pictorial, very neat, and they really helped us understand this complex mechanisms of how the cytokine behaves in in our body, and the importance of the cytokine milieu in deciding the way the body is going to react. Even the last one was a wonderful slide. I think we will remember the talk because of the beautiful slides and the way you presented it. And also helping us explain in detail the mechanisms of atopic dermatitis, the food allergy, and also the JAK-inhibs and the biologics, which are the in thing. So taking us back and helping us understand. So thank you so much for this wonderful talk. Thank you, Dr. Pooja. We are now here to the final talk of this series. As clinicians, one of the most difficult scenarios or one of the most difficult situations that we face is when to order tests and how to interpret this test. Yes, our next expert will talk on laboratory immunology made simple. When to order immunoglobulins, when to order flow cytometries or the LSA, the lymphocyte subsets. And for this, we have Dr. Murli. Dr. Murli Mandakalatur. Sir is an allergist and clinical immunologist at Massachusetts General Hospital in Boston, an assistant professor there. He has vast experience working in this field. I think spanning over 50 decades, something over what my age is also. That is the experience that Sir is going to bring to us today. And it's going to be an honor to learn from him and his vast experience. Sir, may I invite Dr. Murli for this talk, sir?
Thank you. Can you hear me and are my slides visible?
Yes, sir.
Okay.
We can hear you. The slides are visible.
Fantastic. Thank you, Dr. Sinha. At the outset, I want to say all the three speakers have laid a very important groundwork. They talked about cells, they talked about antibodies, cytokines, all of which contribute to the disease process. And when Neeraj Gupta asked me to give this talk, I asked him, what is the schedule for the morning we have? And I think it's beautiful. He's organized it in such a way that we now can take the contributions of cells, antibodies, cytokines to clinical diseases. And as Dr. Sinha nicely pointed out, one of the challenges to a clinician is when you're dealing with a patient with multiple pathways of inflammation or allergic or tumor, what tests do we order? What do these tests signify and how do we utilize them? So with that in mind, let me get started and say, let me see. All right. Why is it not moving? Your screen sharing.
Sir, just double click. Just double click.
Okay, great. Thank you, Niraj. You see, I need your help always. So Niraj has posed these three questions. When to order immunoglobulins, flow cytometry, and subsets. And what I'd like to say is ordering is one thing, but utilizing the test towards a clinical resolution is equally important. So I'm going to try to mix and match both those entities. And as we talked about previously, the antibodies or immunoglobulins are very, very important. And as the picture shows, I'm going to sort of minimize the, okay. As the picture shows, an antibody molecule has both an FAB2 prime, which is the upper half of the molecule, and an FC portion, which was very nicely taught to us by Dr. Mayer. So the FAB region is the one that binds to antigens like bacteria, viruses, fungi, and traps them. They also contribute to the neutralization effect of the toxins. And by binding to an antigen through the FAB end, the antibody creates a phenomenon called opsonization, which means it coats the pathogen, and the remaining portion of the immunoglobulin molecule, the FC portion, can now engage to receptors which recognize this FC portion, and they're called the FC gamma receptors. So the antibody, as I say, works at both ends, FAB prime and the FC. But the FC region has very important pathways of causing subsequent changes in immune defense and immune inflammation. One, it fixes complement via the FC domain, and by activating the complement cascade, it recruits neutrophils, activates them, and helps in killing pathogens. But the most important part we're understanding is how antibody can also be used as a therapeutic molecule, largely because cells like the B cells, the natural killer cells, neutrophils, eosinophils, and monocytes all are endowed with what's called the FC gamma receptor family. And this family includes FC gamma R1, R2, and R3. But in the FC gamma R2, there are two types of receptors. One is called FC gamma R2A, which along with FC gamma R1 and R3 actually help in promoting phagocytosis and elimination of bacteria. But when it comes to FC gamma R2B, which is found on the B cells, antibody binding to this actually turns off the B cells so that you can prevent making autoantibodies. Antibodies binding to FC gamma R2B on mast cells inhibit mast cell activation and basophil activation. So you can clearly see that while the FAB2' is important, the FC regulates the behavior of cells in inflammation and defense. Finally, there's one very important FC receptor called the FC neonatal receptor, which is of great interest to pediatricians because they were the ones who brought the knowledge to the human world. These are found in various cellular linings, especially the vasculature. And what happens is immunoglobulins, as they circulate, bind to the Fc neonatal receptor. They bind to the neonatal receptor, they are prevented from degradation. So what people have used is IVIG in large doses. So if there's a pathogenic antibody and the good IVIG, the IVIG will bind to the Fc neonatal receptors, drive away the pathogenic antibodies from taking refuge or hiding in the vasculature, and then they are degraded. So it alters the kinetics. This phenomenon has been exploited beautifully in the treatment of myasthenia gravis. A lot of studies on multiple sclerosis, and also in chronic inflammatory demyelinating polyneuropathy. And I've given you 3 monoclonal antibodies, efgartigimod, batoclimab, and nipocalimab, all of which modulate the Fc neonatal receptor to inhibit inflammation by getting the pathogenic antibodies degraded. So antibodies are very important. So we're going to see when to choose and how to work on them. Again, this has been very nicely spoken by both the first 2 speakers. IgM is the first responder. It works predominantly in the intravascular compartment. It's a marker of early inflammation, while IgG works in intravascular, extravascular, and interstitial compartment, and it modulates various cellular functions. And IgA is the mucosal guardian. It protects the mucosa. It's also a tolerogen by inducing tolerance. And IgA, as we have talked about, protects against parasites, and it's also triggered by alarmins, which has been alluded to. The important part I want to recognize is that intravenous immunoglobulin, as we give it to patients, helps opsonization. It also modulates pharmacokinetics of endogenous immunoglobulins, and particularly if they're pathogenic, the Fc neonatal receptor will facilitate their egress into the vasculature, and it will degrade them. Most importantly, IVIG can also work on Fc gamma R2b and inhibit autoantibody, and that's one of the reasons they have been used in autoimmune hemolytic anemias. So while the natural immunoglobulins do play a role, it's very, very important to recognize that monoclonal antibodies have beneficially altered our therapeutic landscape. So with that as in mind, when do we order immunoglobulins? And as was very nicely pointed out by Omer, immunodeficiency diseases are very, very important first reason to order that. And he also pointed out the entity called transient hypogammaglobulinemia of infancy, which occurs somewhere between 6 months and 24 months by the time the baby's immunoglobulin takes over and the mother's wanes away. And in some children, the development delay happens, especially if they are what they call children who are born before the period of gestation, and where their immune maturation slows down. So they have a period of hypogammaglobulinemia. So by sequential measurement of IgG, A, and M, you can understand what's happening. And the moment the IgG starts going up, the child is recovering from transient hypogammaglobulinemia of infancy. The other important areas in the immunodeficiency landscape are very well discussed. Again, anything that's involving the enzymes or genes, right up to the development of a double positive B cell, which is IgM and IgD. And though the immunoglobulin measurements G, A, and M are very low, but it can also be low in an acquired immunodeficiency. But the important part is examine the patient. If there are no tonsils, no palpable lymph nodes, and the spleen is small, then you are dealing with the whole syndrome, which is very nicely described, starting from ICAROS enzymes to glutathione tyrosine kinase to ICOS ligand deficiency and CVID. On the other hand, once you get a B cell switch and the syndrome of hyper IgM syndromes, which is again pointed out due to CD40 ligand, UNG, etc., the M is high, the G and A are low. So when you're looking at a child with a recurrent immunodeficiency, it's important to look at the G, A, and M. If all the 3 isotypes are low and no tonsils and no lymph nodes, then you're dealing with a congenital immunodeficiency, which is different when enlarged lymph nodes and hepatosplenomegaly is present, which is a hyper IgM syndrome. So in measuring immunoglobulins, in B cell developmental disorders is very helpful. They're also helpful in combined immunodeficiency, severe combined immunodeficiency, patients with thymomas, CVID, and a very important entity called IFEC syndrome, where you get immunodysregulation, polyendocrinopathy, um, enteropathy on an X-linked gene. In this syndrome, what you will find is very low G, A, and M because they lose a lot of proteins via the gut. So hypogammaglobulinemia with or without hyper IgM can be very useful. On the other hand, immunodeficiencies can present with increased immunoglobulins. Particularly, you're looking at some of the innate immune disorders or proliferative disorders. Chronic granulomatous disease, which is a disorder of innate immunity, lymphocyte adhesion defects type 1, 2, and 3, hemophagocytic lymphohistiocytosis. In all these conditions, the innate immunity fails. The adaptive immunity tries to compensate by making a lot of antibodies. So you can get somebody with recurrent infections, skin nodules, Aspergillus disease in the lung as part of CGD, in whom the G, A, and M is high. So this is an attempted compensation. And then you must always see in somebody who has cold abscesses, staphylococcal syndromes, big lung abscesses, which lead with the pneumatococcus. Think of the hyper IgE syndrome, along with cutaneous manifestations. Here, the IgE is very high. So measuring immunoglobulins, G, A, M, D, and E will be helpful in immunodeficiency diseases. The second group is allergic disorders. If we look at a common, simple seasonal allergic rhinitis or even food allergy, the immunoglobulins are all essentially normal. The E may or may not be elevated. Sometimes the skin tests are positive. The serum IgE is normal because the preferential home for IgE is binding to cells. Once they exceed a certain threshold of production, then the serum IgE goes up. So you can't make a diagnosis with you can make a diagnosis of allergy with a normal serum IgE. Skin tests are exceedingly important clinical correlation. The chronic rhinosinusitis with or without nasal polyps depends on the state. Early on, the immunoglobulins will be normal, including IgE, but as the disease progresses and the B cells get activated, you not only get IgE elevation, but the other isotypes are elevated. So acute, they may be normal. Chronic recurrent, they're often elevated. And in atopic dermatitis is the same stage. If you see an acute atopic dermatitis, IgG and M are normal, but E is almost always elevated due to the atopic diathesis. Once they get chronic disease, the IgG becomes elevated beyond the normal. That's important. The one disease which is very common in warm, humid countries is allergic bronchopulmonary aspergillosis. And they can exacerbate and remit. And here, immunoglobulins are very, very important. In exacerbation, aspergillus-specific IgG goes up. And when you treat, the levels drop. IgA is always elevated because it's a mucosal immune response in the lung. But the important thing is also to look at IgE. If the E is very high, you treat and the E drops along with allergen-specific or aspergillus-specific IgE, the patient is entering into a remission. IgM may or may not help, unlike chronic bacterial infections. So using IgE and IgG levels help you in detecting exacerbation versus remission and helps in managing patients with ABPA. The other area where immunoglobulins are very helpful for allergists and immunologists is systemic infections. In acute infection, IgM goes up, as was very nicely taught to us by Umair. But in chronic infections, the M often comes down because of the class switch and the IgG gets elevated. But this IgG is polyclonal. And in pediatrics, it's not so important, but in adult medicine, patients with myeloma, patients with lymphoma can come with the elevated IgG. But that on SPEP, serum protein electrophoresis, is monoclonal. So whenever you get an elevated IgG in an infective state, if it's adult, you must do an SPEP to qualify what's the nature of the proliferation. In pediatrics, it's almost always polyclonal. Then in parasitic diseases, IgE is very helpful and important. So among a group of gastrointestinal disorders, it's very important to look at malabsorption due to celiac or intestinal lymphangiectasia, The gut leaks all the immunoglobulins, everything is low, including IgE. In an entity which is becoming very increasingly recognized as metabolic deficiency associated steatohepatitis, often called fatty liver, or in cirrhosis, the IgA is increased because of the mucosal stimulation. The IgG is also increased, and you get a very characteristic appearance on SPEP called beta gamma bridging. But in this disorder, early on is increased, later on the IgG takes over class switch. F5's food protein induced enterocolitis syndrome is very tricky. During acute phase, the GA and M decrease, but the IgE is not elevated because it's not an IgE mediated disease. And in some patients, it could be borderline if they are atopic, but this is not an allergic mechanism. The next group of diseases we need to look at is autoimmune diseases, where there's chronic antigen stimulation, autoantibody production, and the troublesome autoantibodies are almost always of the IgG variety. They're all increased, but it's polyclonal. But there's one autoimmune disease which masquerades with eosinophilic syndrome, which could be mistaken for eosinophilic pneumonia, which could be mistaken for allergic bronchopulmonary aspergillosis, is eosinophilic granulomatosis with polyangiitis, the old Churg-Strauss syndrome. Here, it's very, very important. Unlike microscopic polyangiitis or the granulomatous polyangiitis, the IgE is elevated. EGPA is unique in that it's got an hematopoietic as well as vasculitic component. So IgE is very useful in discriminating for rheumatologists when they're dealing with autoimmune diseases. Tumors are known to be greatly immunosuppressive, especially solid tumors, so all the isotypes can be normal or decreased. But in lymphomas, IgE is often elevated. So the patient may come with a rash and elevated IgE and a referral, and if he has lymph nodes, you've got to have lymphoma and differential diagnosis. I already talked about monoclonal B cell proliferative disorders. If it's IgG proliferating cell, the IgG is increased, but always the bad guy shuts off the good guys, the A and M are decreased. So you always look at the balance of the isotypes, and IgE is almost always normal. But it's important when the G is elevated and you suspect a multiple myeloma in somebody with urticaria, or if somebody comes with purpura and you think they have Waldenstrom's, when you get the elevated G or M, especially in the adult population, you must do an SPEP and immunofixation, and in these days also serum-free light chains. So that's something the average allergist doesn't see, though once in a while we get caught. Finally, monitoring response to therapy. When you give IVIG, you need to see how much you're giving. Is it adequate? What is the good threshold? So IgG alone is adequate because immunoglobulin intravenous does not contain MRA. So follow-ups can be done with IgG, but the initial evaluation is GA and N. Then whenever you treat them with FC neonatal receptor monoclonal antibody therapy, you got to monitor the IgG levels. Finally, as was very nicely talked about, the role of checkpoint inhibitors, graft versus host disease. In these situations, you have to have a baseline GA and M because you got to monitor therapy. And if the G drops at a critical level, you replace with IVIG and you monitor them with sequential IgG, A, and M. So immunoglobulin determinations are very, very helpful. The second part of the challenge, which Neeraj told me, is talk about flow cytometry. This is one of the most powerful techniques in immunology. It has actually opened up the entire diagnostic armamentarium. It's very simple. There are three things you need to know. One is you have to have cells from a patient you want to analyze. Could be a CVID, could be a congenital immunodeficiency, could be somebody with a nasal polyps where the tissue has been digested. The cells are labeled with a fluorescent antibody that you're interested. For example, you want to study CD3 and CD4 cells, you label with CD3 and CD4. If you're interested in looking at a patient with eosinophilia, you can stain for a surface marker called EG2. Or if it's a basophil, you can stain for CD63. So you label the cells you want to analyze. The cells come down and they're driven into a single file by fluid in the sheath. So these are like soldiers marching down the aisle, and they are being examined. And you hit a laser beam. The laser beam looks at two things. What is the scattering of the light as it hits the cell? And then what is the fluorescent marker which is ignited or excited? So let's look at this. So when the beam of light comes, you get a big cell, you get a big forward scatter. So granulocytes are large, so they are forward scattered. Lymphocytes are small, so they are left forward scattered. So forward scatter is dependent on the size of the cell. The internal complexity, the granules in the nuclear structure determine the side scatter. Again, neutrophils are a lot of granules, so they're not really large, they're complex in their biology. Monocytes are both the same size, but they have less secretory granules. And lymphocytes have a lot of potential to synthesize cytokines, but they don't have granular structure. So they are much smaller, and so you can gate them. So you put your gate here, you can study granulocytes, and in that are the basophils for basophil activation test. You want to study lymphocytes, you gate them. So what immunologists do is they first gate the cell they want to study. Then they stain them. And what is the principle? You got 4 different kinds of cells. The green has antigen A, the B has the brown has antigen B, the yellow cell has both A and B, and the dark cell has neither. So you stain with appropriate antibodies, send them through a single file, the laser ignites, and you get the intensity of whatever you were studying on one axis, and this is dual color. So take a look. So based on the intensity, this is what happens. So for example, let's apply this to CD4 and CD8. So you stain on the x-axis for CD4, and you stain on the y-axis for CD8, and let us say that is what you want to exploit in this bloodstream in a patient. So they will segregate based on the colors to 4 quadrants. If a cell has both A and B, it's yellow, and that will be your cell which is CD4 positive and CD8 positive. So this, as you go from left to right, the intensity increases, the cell lights up. Then when you come down to the green cell, the green cell has CD4, but CD8 is negative. So you can now identify CD4 plus CD8 minus cells. All the TH cells, which Pooja talked about, TH1, TH2 are all in that lineage. Then for the oncologist, they want CD8. So here you see the CD8 is positive, CD4 is negative, and obviously the cell which expresses no 4 or 8, like a double negative cells entering the thymus, that's there. So this is how flow cytometry can be used to analyze it. The other condition which is important from innate immunity is chronic granulomatous disease. And there are at least 3 or 4 very good cases from India which came to our institution for analysis. And they all had CGD of various types. First of all, how is this test done? You take neutrophils, you activate it with a stimulus, such as formol myristate acetate. And then you load the neutrophils with a red dye called rhodamine. When the rhodamine, when it's not activated, is colorless. When the cells make superoxide or hydrogen peroxide, which means they have the phagocytic oxidase pathway, they convert dihydrorhodamine into rhodamine, and you see how the axis shifts in fluorescence. So this is a control cell. When you activate it, it shifts to the right. A patient with CGD, you load it with dihydrorhodamine, you stimulate it, the unstimulated cells are like that. When you stimulate, the patients don't make superoxide, don't make hydrogen peroxide, they cannot make reactive oxygen species, they do not change the color of rhodamine, and they don't shift. If the mother is a carrier, because it's X-linked, so she has a bad gene and a good gene. If this gene is transferred to the child gets CGD, but if this gene gets transferred and the dad is normal, you get a healthy child. So flow cytometry is useful for analyzing adaptive as well as innate immunity. In the field of allergy, something which is close to Neeraj's heart, and we've got to get this going, is basophil activation test. I'll give you the principle very quickly. there are two important CD markers. 203 is on the membrane of unactivated basophils. CD63 is inside. If you take the B cell, a basophil and activate it by an allergen binds to IgE, cross-links, activates MAP kinase, phosphorylates. CD23 is upregulated. It's already there, but it's increasing. But CD63 is now makes its appearance on the membrane. So you can take that advantage and do what's called basophil activation test. Basophils are HLA-DR positive and they are CD123. So you gate it on the neutrophils and pull out the basophils. Then you see when you activate it with nothing, with the media, the gray triangle, you're not activated. When you put anti-IgE, the basophil 203 goes up, 63 really goes up. And that's basophil activation. Now you can use this for diagnosing food allergy. You use components to activate it. You can diagnose venom specific when you can't do skin test. For example, in a patient with peanut allergy, the gray is unstimulated. And when you take the basophils, add the peanut component in the patient's cells and serum, you can see how it shifts to the right. And that CD63 is unleashed, saying that the patient's basophils are activated by allergen. And that's CD63. It's also very useful in analyzing mastocytosis, which is very important differential diagnosis for idiopathic anaphylaxis. Flow cytometry is excellent. You can do it on peripheral blood if you're lucky, otherwise it's always done in the bone marrow. Take a look. Mast cells have a molecule called c-KIT, which is CD117, and they are also IL-2 receptor positive. So you put it through a flow cytometer. This is somebody who has mast cells, but they are not activated. So that's not mastocytosis. But if the mast cells also express CD25, that's abnormal. They also express CD2. So you can do three colors, CD2, CD117, and CD25, and if they're positive, that's a syndrome of mastocytosis. You do not see that in anaphylaxis. So I'm not going to go into details, but to say you can use flow cytometry and analyze a variety of lymphocyte subsets. You can study infectious disease, and the first disease where CD4 cells by flow cytometry to analyze CD4 and lymphocyte subsets was HIV disease. But we can also take the same principle of flow cytometry and study a panel of T cell antigens to dissect immunodeficiency. For example, if somebody has a severe combined immunodeficiency, there'll be CD3 negative, CD2 negative, and CD19 negative, which means they don't have T and B cells. But if there is this CVID, they often have T cells and their B cells may be normal, but their specific antibody response is negative. So by exploiting T cell markers, B cell markers, and NK cell markers, one can use lymphocyte subsets to diagnose it. Also very important is you can use flow cytometry to diagnose LAD1 and 2, because in LAD1, CD18 is deficient. In LAD2, CD15 is deficient. Lastly, how does flow cytometry help? You and I as clinicians are often challenged by patients with pruritus, urticaria, erythroderma, and severe eczematous skin. And this could be idiopathic hypereosinophilic syndrome, it could be eczema, it could be T cell lymphoma. When you do a flow cytometry, you can really sort it out. In patients with T cell lymphoma with eosinophilic variant, the abnormal T cell, which makes both IgE and eosinophilia by cytokines IL-4 and 5. So on the x-axis, you stain for CD4, and on the y-axis, you stain for CD3. These patients have a very unusual phenotype. They are CD3 negative, but CD4 positive. So we learn in medical school that all T cells are CD3 positive, but mutant cells don't read the textbooks. And so what you have is when you do a flow cytometry, you see you get a CD4 cell, but it's CD3 negative. If you see that on flow and on the lymphocyte subset, then you can clearly say this is a T cell lymphoma, very different treatment from idiopathic hypereosinophilic syndrome and certainly from severe eczema. So it's very, very helpful. So what I have done is combine flow cytometry and lymphocyte subsets because they work hand in hand. But the important take home message is lab tests not only help us to facilitate diagnosis, rule out the masqueraders, monitor disease activity like CD4 counts in HIV, make therapeutic decisions when to give IV gamma globulin, when to treat an ABPA, which is exacerbating, and also recognize untoward side effects. Important to recognize that the consequences of immune responses are reflected in immunoglobulin levels, T and B cell numbers and function, and many of these are measured by flow cytometry, employing lymphocyte subset biology. Lastly, something we haven't touched on and is probably going to come in the next 5 years. When we talk about a CD4 cell, it could be Th1, Th2, Th17, Treg. Well, right now, we don't have the ability in a commercial clinical lab to study intracellular molecules. If the CD4 cell has inside IL-4 and IL-5 genome, that's a Th2. If it has IL-18 and IL-12, it's a Th1. So the future is to stain cells, look at intracellular flow cytometry, coupled with cellular markers, and understand the biology. And this will also enhance therapeutics. So I'm going to stop here by saying I want to thank you all for listening. And besides Dashara, today is Mahatma Gandhi's birthday. And yes, we all have to conquer evil, establish goodness, but it should be a world of peace. And there's no better person who brought a whole nation without bloodshed, without prolonged war, and got independence, which we all very cherish. And so today is also Mahatma Gandhi's birthday. I'm going to stop here.
Thank you, sir. Thank you for this excellent and detailed talk on the lab tests for immunology. I know we are, it's already 10, but allergy and immunology is a subject or uh, is a field where we have a lot of whys and a lot of questions, and it is still evolving. We all are trying to understand it. So I don't want to miss out on this opportunity where I have experts on this subject from all fields with such vast experience and researchers. So I just have maybe just one or two quick questions on all the whys of allergy and immunology. Sir, you said that following IVIG replacement, we generally can measure the IVIG levels to see whether they are adequate. But sometimes in patients with immunodeficiencies or inborn errors of immunity, there are a few cases when the when the IgG levels are normal or near normal, but still IVIG replacement helps them. So, how does that happen?
All right.
And
So
when do
there
we
is no
get...
magic level. There's no magic level for IgG. Some people can have an IgG of 300 and be completely healthy. Somebody can have an IgG of 600 and get recurrent infections. So what you have to do is use that personal best. In other words, you treat the patient IVIG, gets infections, increase the dose, increase the frequency. Let's say they reach a nadir, a pre-infusion of 700 milligrams. That means 700 milligrams and above is the threshold. So you don't treat a magic number, but you got to identify the protective threshold in that patient. And then you give IVIG to make sure it doesn't drop below that level.
Okay.
So
Okay.
there's no magic number. It's the functional ability of the immune system to utilize the IV gamma globulin. And that's IgG, A and M are not in the preparation.
Okay. You spoke about the future, the intracellular signaling molecules, and how that is the future. I just want to know that allergen immunotherapy is something that we all are very excited and we offer to our patients also. But beyond allergen immunotherapy, are there any other new novel immunotherapeutic strategies that are being worked up in or being researched upon for something like a permanent cure or, permanent how do we outgrow allergy? Is something being worked on that field?
Well, that's all in the field of what's called tolerance. How do you induce tolerance to a specific antigen? That is the holy grail of immunology. We have not reached that goal as yet. But there are some very interesting molecules. Along with allergen immunotherapy, trials are going on where they take the allergen and couple it with a, let's say, peanut specific IgG. So this IgG binds to the mast cell Fc gamma R2b as well as activates it. So when you activate the cell and inhibit it, the cell goes into paralysis. It doesn't make IgE. So one way of trying to induce tolerance is to activate and inhibit at the same time, create a state of confusion in the intracellular machinery of the mast cell, so the mast cell doesn't continue to make histamine leukotrienes. So this is being tried at UCLA. It is still in clinical trials, but it's quite promising. So they are called bifunctional antibodies.
They are called bifunctional.
Bifunctional, because they bind to the Fc gamma R2b and they also interfere with the IgE signaling. So they act at two levels.
Very interesting. Very interesting. Actually, immunology as a whole is extremely interesting, the way it works. Again, coming to another why, allergen immunotherapy, we call it is the vaccine of allergic diseases. That is, many times that is how we tell our patients. But then the patients have a question that when you vaccinate a child, you nearly have the child does not get the disease again. But once we stop allergen immunotherapy, it many times it tends to come back. Sometimes in just a year or so, we are back with the disease. So why should that happen, or how can we outgrow that?
Right. So I think someone there talked about trained immunity. So that is where we need to go. Our allergen immunotherapy is capable of causing a tolerance temporarily. The moment you stop, you see the allergen is also there. So the enemy is there, the protective function is there. But when it comes to pneumococcal pneumonia, when you vaccinate them, you not only induce B cells, you use memory B cells, and there are T cell memories. And we don't have pneumococci rampant every day. So the state of antibody protection lasts much longer. But in allergens, the underlying genetic diathesis, whether it's a leaky basement membrane or filaggrin deficiency in atopic dermatitis or abnormal mutations of the IL-4, IL-5 gene cluster, that defect is still there. So you have to counteract it with allergen immunotherapy. It also says that allergen immunotherapy at this stage is like a passive antibody therapy. What we need is something very active, and that is inducing tolerogen. I don't think we have come to that far. It will probably be an understanding of mucosal tolerance and how to induce tolerance. Great questions. It only says the field has to move ahead.
Yeah. Just one quick question for Dr. Pooja. You spoke about the cytokine milieu and how that actually is it is very important in the way the body is going to react. And you also spoke about the environment and many other things that can this. Just something that is there something in our lifestyle or anything that we can do which can just drastically shift the cytokine milieu in the positive side, or something that would avoid. Uh-huh. I know these are all wise, and that is what I said, that I'm just taking this opportunity to just ask and dig deep into this topic, which is which has so many mysteries behind, and the mysteries need to be unlocked. So, but all in all, a wonderful talk. Something you wanted to say?
If we can modulate our epithelial barrier, I think for the allergy part, for type 2 immunity, probably that is one of the main triggers. Although that is not the only one, there are others. Still, like to totally avoid soap and all those is difficult.
Yeah.
Some balance needs to be there. No one answer for that.
I think it was an wonderful sessions. the fundamentals, the basics, but even how does that actually impact allergy and immunology. It's been superb learning, many new things that has come up. And I really thank all our speakers, Dr. Samajda, Dr. Umair, Dr. Pooja, and Dr. Murli sir, for being with us here. A very big thank you to all of you all for kickstarting this webinar series on applied immunology. Thank you so much for joining. Now I hand over to Dr. Neeraj for the to conclude the session.
Thank you, Dr. Mangai. I think you rightly pointed out that fantastic sessions, and definitely this session needs to be reviewed and replayed again and again so that we can understand it more and more. And the session is available on the YouTube channel, so anybody can go there, consult once again, and then review, rebrush itself. Definitely, it will be needed. And excellent moderation by you. So now, in continuity of that, tomorrow we'll be discussing about the I will say the immunological basis of all different type of allergies. That could be respiratory allergy, could be food allergy, could be anaphylaxis, and simultaneously immunological basis of immunotherapy, which is, I think, very much essential, very much needed. So from the allergist point of view, after today's foundation, tomorrow's talk will be, you can say, like mandatory talk to attend for every allergist. So thank you very much. Thank you. And I would once again like to say thank you to everyone for contributing so fantastically today. Thank you. Thank you.
Thank you, everyone. Thank you.
Thanks, Neeraj. Thanks, everybody.